Video we're going to talk about malaria, and more specifically we're going to talk about Plasmodium and its life cycle. So Plasmodium is a protozoa. It is a protozoa and its class is trypanosoma. Alright, so Plasmodium, how does it look like? Plasmodium looks like this, with a lot of cilia. This is Plasmodium, with two vacuoles, one big vacuole central, and then peripheral nucleus, and then we have the a smaller vacuole, and a lot of cilia. So that's how it looks like. The vector and the definitive host is the Anopheles mosquito, which is female. It's important to know that it's only female. Mostly mosquitoes are vegetarian, but the female mosquitoes, which are mostly pregnant, they need blood of humans for maturation of eggs. Okay, so that's important to know. So because of this, there are two phases for the life cycle of Plasmodium. One is the sexual phase and the other is the asexual phase. The sexual phase is known as sporogony, and the asexual phase is called as schizogeny. So here is a big overview of the life cycle that I took from Livingston's microbiology book. And we're gonna go through this in a bit of an overview or a helicopter reading. So we are gonna start with the stage A of the asexual cycle, that is the schizogeny in the humans. So the schizogeny occurs in the humans. This occurs in the humans, and this occurs in the mosquito. Okay, that's important to know. Now what is going to happen is that a mosquito right here is going to take a blood meal, and this is going to inject sporozytes. How these sporozytes are formed, I'm going to tell you when we talk about the mosquito stage. These sporozytes are then, they travel to the liver cells where everything is detoxified. The blood is detoxified in the liver. That's how it reaches the liver. It infects the liver cell, the sporozytes, and then it is going to form a schizont, a big sac filled with a lot of merozytes. That is a schizont, right? So this is a big sac, right? This is the whole cell. This is the whole cell right here. Inside the whole cell, there is a big sac-like structure you see right here. That's the schizont. In this schizont, we have merozytes. The schizont is going to then rupture and release the merozytes. These merozytes are released into the blood. Then we enter the stage B. That's the erythrocytic cycle. Here, the merozytes are going to then enter the human blood cells, the RBCs. Then from the RBCs, it is then going to form an immature trophozoid, which is a ring-shaped structure. Okay, you see this ring-shaped structure. That is a trophozoid. Now, essentially, two things can happen. This mature trophozoid then can become schizont, or this trophozoid is going to form gametocytes. These are two things that can happen. If it becomes a schizont, then this schizont is going to rupture and release more schizonts, and then it is going to reinfect the human blood. And that is how the malarial symptoms that I'm going to also explain a little bit in further occurs, because of over and over again the infection of the red blood cells by schizonts and the merozytes. If it forms gametocytes, there are two gametocytes. male and female, macro and micro. So, because the Plasmodium has 4 species, the Falciparum, vivax, ovale, and malaria, all of them are mostly the same. There are some points that you need to remember. I'm going to tell you a little bit later in this video. And these gametocytes are now going to get into a non-infective mosquito, a different mosquito. It is going to take a blood meal and it is going to ingest these gametocytes. Now this mosquito, inside this mosquito, we have a macrogametocyte and a microgametocyte. The microgametocyte is going to enter the macrogamete and it is going to then form a ookinete, which is basically motile. This is motile. As this is motile, it is now going to enter the intestine, the gut, the midgut of the mosquito, and it is going to form a oocyst. And then this oocyst is going to rupture and release sporocytes. And then the cycle continues. Now you know how sporocytes are formed. So sporocytes are basically the spores that is going to form more and more plasmodia. That is the life cycle of malaria. Sorry, the Plasmodium. Now, pathogenesis. Of course, I told you the release of merocytes and also sequestration of infected RBCs by spleen is going to cause RBC damage. And this RBC damage is going to cause hemorrhage and necrosis. Now, these can also go into the brain and cause cerebral malaria, and it can also go to the kidneys and cause kidney damage and hemoglobinuria and acute renal failure. And because of a lot of hemoglobin in the urine is going to get dark, and hence it is called as dark water fever. And because spleen is doing a lot of job for sequestration of infected RBCs, we're going to have splenomegaly. Alright, now some points that you need to remember. Plasmodium malaria, the species malaria, it has a 72-hour fever cycle, which means that after 72 hours, like day 1, and after like 4 days, like after 4 days interval, we're gonna have a spike of fever, like high grade fever. Then the fever is gonna go back to normal for 4 days, and then we're gonna have another spike of fever, and that is called as quartron. And other plasmodia like ovale, falciparum, etc., they have a tertian fever cycle, which means every 3rd day with a gap of 3 days, right? Now, other points are Plasmodium falciparum has the most increased parasitemia. This is going to be very much infective, most infective of all, and it is also chloroquine resistant, which chloroquine is a drug that is used to treat malaria. Plasmodium vivax, it is only going to infect reticulocytes. Plasmodium malariae, it only affects mature RBCs. Reticulocytes are immature RBCs, and mature RBCs are infected by malaria. And lastly, you just need to know at the back of your hand is that sickle cell anemia patients and G6PD patients are actually protected against malaria because their blood cells are not, they cannot harbor the merocytes. That's why they cannot form gametocytes. They cannot form the merocytes. As I told you, blood cells are important for the life cycle of Plasmodia. If you have a sickle cell, it is not going to work. It won't harbor a trophocyte, and that is it.